Welcome to The Explainer. Have you ever wondered how a simple thought, a fleeting memory, actually works? Well, today we're going to build a complete picture of just that. We're talking about how neurons communicate, from the basic electrical signals all the way to the advanced lab techniques scientists use to study them.
You know, at the very center of all brain function is this place right here, the synapse. It's this tiny, tiny gap where one neuron sends a signal to another one. And it does this using these chemical messengers we call neurotransmitters. Everything happens right here in this little space.
So we're going to build our understanding from the ground up. We'll start with the neuron when it's just sitting there at rest. Then we'll look at the signal itself, the action potential. After that, we'll see how that signal crosses the synapse. And finally, we'll take a look at how scientists can actually watch all of these incredible processes happen. Okay, let's dive into this.
Right, so before a neuron can fire off a signal, it has to establish a kind of baseline electrical charge across its membrane. We call this the resting potential. Think of it as a battery, charged up and ready to go.
Now, this potential is caused by a really specific imbalance of ions. If you look at this table, you'll see what I mean. Notice the super high concentration of potassium, that's K+, inside the neuron. And then see all that sodium, Na+, that's mostly on the outside? This separation is absolutely crucial.
So how does this resting potential actually happen? Well, first, you've got this massive concentration of potassium ions inside the cell. Second, the cell membrane is selectively permeable. It has these little channels that are always open, but pretty much only for potassium. So, potassium ions start to leak out. And as these positive ions leave, the inside of the cell gets more and more negative. This creates an electrical charge that starts to pull the positive potassium ions back in, and eventually, you reach an equilibrium. The chemical force pushing potassium out is perfectly balanced by the electrical force pulling it back in.
Okay, so while that ion leakage is what creates the resting potential from moment to moment, there's another player that maintains the whole system in the long run, and that's the sodium-potassium pump. This thing is a little machine that actively pumps 3 sodium ions out of the cell for every 2 potassium ions it pumps in. It uses a ton of energy, but it's what keeps those concentration gradients stable.
Let's move on and see how this all builds into the main signal of the nervous system, the action potential. A lot of what we know here comes from some pioneering work by Hodgkin and Huxley back in 1939. And they figured this out using giant squid axons of all things. They discover that this signal, the action potential, is this brief all-or-nothing reversal of that resting potential. It flips to inside positive for a split second and then travels all the way down the axon without fading out or decreasing in size at all.
So what makes this reversal happen? It's all orchestrated by voltage-gated ion channels. Now, the sodium channels are a bit complex. They have 3 states. They can be closed, they can be open and activated, or they can be closed and inactivated, which is a really important distinction. The potassium channels, on the other hand, are much simpler. They're just either closed or open.
Now, what's really cool about this slide is how it shows you the exact sequence of events. That green line, the membrane potential, shoots up as the fast-acting sodium channels, that's the blue curve, fly open. Then, almost immediately, those sodium channels inactivate and the slower potassium channels, the orange curve, start to open. That causes the potential to fall back down, repolarizing the cell. It's a beautiful, precise dance.
And this illustrates brilliantly how that signal actually travels. In an unmyelinated axon, it's just a slow, continuous wave. But in myelinated axons, the signal literally jumps between these gaps in the myelin sheath, which are called the nodes of Ranvier. This process is known as saltatory conduction, and it is dramatically faster.
Okay, so now we understand the signal within a single neuron. But let's get to the really interesting part, how it gets passed on to the next cell at the synapse.
The whole process is a very precise 5-step sequence. First, the action potential arrives at the nerve terminal. This triggers voltage-gated calcium channels to open. The flood of calcium causes little bubbles full of neurotransmitters, called vesicles, to fuse with the membrane. This fusion releases the neurotransmitter into that tiny gap, the synaptic cleft. And finally, those neurotransmitter molecules bind to receptors on the postsynaptic neuron, passing the message along.
Now, the chemical signals themselves are incredibly diverse. They fall into several major classes. You've got your biogenic amines, which include things like serotonin and dopamine that you've probably heard of. Then you have amino acids like glutamate and GABA, peptides, and even other small molecules like ATP and nitric oxide. Each one has a different job.
And you know, it's not just about the neurons. We have to talk about glial cells. They're the critical support staff.
For example, oligodendrocytes in the central nervous system provide that myelin for saltatory conduction. Fun fact, their membranes also contain a protein called NOGO that actually inhibits nerve growth. And then you have astrocytes, which support the whole system and buffer the ion concentrations outside the cells, making sure everything is just right for the neurons to work properly.
So, how in the world do we know all of this? Let's look at some of the foundational techniques that neuroscientists use to actually study what's happening at the synapse. First up is a really key tool called the synaptosome. This is essentially an isolated, intact nerve terminal. What happens is, when you homogenize brain tissue, the presynaptic terminals actually tear off and then reseal themselves. So you end up with this little packet that has everything you need for neurotransmission, perfect for studying in a test. To isolate these synaptosomes, you first homogenize the brain tissue, and then you use something called differential centrifugation. It's a two-step process. First, there's a slow spin, at about 1,000 times the force of gravity for 10 minutes. Then, you take the liquid on top, the supernatant, and spin it again, but this time, much faster, at 10,000 G for 20 minutes. This pellets the synaptosomes, concentrating them at the bottom. And for even more purification, you can use a sucrose gradient. When you spin the sample in this gradient, all the different components separate out by their density. The synaptosomes settle into a distinct layer, neatly sandwiched between the lighter myelin and the heavier mitochondria. It's a really elegant way to get a pure sample. All right, let's move on to some of the more advanced methods, including things like stem cells and actually tracking transport inside the axon itself.
This is just incredible. Scientists can now create neurons from a patient's own somatic cells, like skin cells. They do it by adding a cocktail of just 4 key transcription factors. This process creates what are called induced pluripotent stem cells, or iPSCs for short. And of course, these reprogrammed cells have to be validated to make sure they're the real deal. Scientists confirm that they share all the essential characteristics of embryonic stem cells, everything from their appearance and how they grow, to their gene expression, their epigenetic state, and their pluripotency.
Now, inside the neuron, materials are constantly on the move. And this table shows you the different rates of this axonal transport. You've got fast transports moving vesicles along at 400 millimeters per day, all the way down to slow transport for things like cytoskeletal elements, which move at less than 1 millimeter per day. And this transport is an active process, driven by what we call molecular motors. There's a motor called kinesin that walks materials like synaptic vesicles and mitochondria away from the cell body. And then there's another motor called dynein that's responsible for retrograde transport, moving things back toward the nucleus. It's this constant, active process that keeps the neuron alive. So as we continue to refine all these amazing techniques, you just have to wonder, what fundamental questions about the brain will we finally be able to answer next?